Okay, the final process of breathing or respiration that I want to talk about is just the transport of respiratory gases. We already talked a lot about the cardiovascular system, but let's focus now on the transport of oxygen and carbon dioxide. So we're going to start with oxygen. Remember when we talked about the solubilities and Henry's law? Oxygen has a really low solubility in water. So we wouldn't expect to find a lot of oxygen dissolved in plasma, which is predominantly water. So when we talk about transport of oxygen in the blood, we find only about 2% of it dissolved in the plasma itself, because it has a low solubility. So because it has a low solubility, it has to find a different route for transport, and it has solved that by binding to hemoglobin inside of our red blood cells, specifically to an iron atom on one of the globulin chains of hemoglobin. So 98%, or the vast majority of our oxygen, is transported via hemoglobin. When we have oxygen bound to our hemoglobin, we call it oxyhemoglobin and represent it with Hb, which represents hemoglobin, with molecular oxygen O2. Another thing I want to talk about when we talk about binding, it doesn't matter if we're talking about oxygen or carbon dioxide. When either one of these bind hemoglobin, they're going to change the shape of hemoglobin. And when you change the shape of hemoglobin or any protein, it's going to change its affinity for something else, meaning its willingness to bind something else. So once we bind that first molecular oxygen to one of the globulin chains in hemoglobin, we are going to change the shape in hemoglobin in such a way to increase its affinity for oxygen. So once the first one binds, it's more likely to bind more oxygen. Factors that influence are oxygen loading and unloading from our hemoglobin, partial pressure of oxygen, so that's definitely going to influence it. The higher partial pressure means the higher oxygen levels we have in our blood, the more likely we're going to have oxygen bound to hemoglobin. Then we have some other factors, which we talked about in lecture: temperature, the partial pressure of carbon dioxide, of course, a hydrogen ion, and something called BPG. All of these are byproducts of metabolism within any cell. So if we increase any of these, they're going to cause a decrease in hemoglobin's affinity for oxygen. Okay, which means they're going to increase oxygen unloading or being unloaded from hemoglobin, because we've lowered how much hemoglobin actually is attracted to oxygen. We'll talk about this in a second, how this happens. So again, all of these are byproducts of metabolism. They're increased in response to metabolism, or the consumption of oxygen, and they're going to decrease the affinity for oxygen to bind hemoglobin, which is going to enhance removing oxygen from hemoglobin. So because all of these would do that, where would we expect the levels of all of these to be the highest? We would expect all of these to be the highest in systemic capillaries. Why? Because oxygen unloading is the goal in systemic capillaries. So we picked up in our lungs our oxygen. We have our oxygen bound to hemoglobin, 98% of it. Now it's being transported via the cardiovascular system to try to be delivered to tissues that have used a lot of oxygen, and now they need more oxygen. So because we want to deliver the oxygen from our bloodstream and our systemic capillaries to our tissues, we want oxygen to want to come off of hemoglobin. We want to lower that affinity of hemoglobin towards oxygen. So, in the systemic capillaries where we're reaching our tissues and oxygen unloading is the goal, we would expect to have the highest level of all of these: a slightly increased temperature, a higher partial pressure of carbon dioxide, a higher hydrogen ion concentration, as well as another chemical byproduct called BPG. So that brings us to what's called the Bohr effect. So basically, let me change colors. The Bohr effect is that as we increase carbon dioxide, we're going to also increase the hydrogen ion concentration. And we'll talk about that with the bicarbonate ion in a minute. But if we increase the carbon dioxide concentration, we increase the hydrogen ion concentration, which means that we've increased the acidity of something. So the greater the hydrogen ion concentration means the greater the acidity, and the greater acidity of something means that it has a lower pH. So anytime we have an increase in carbon dioxide or hydrogen ion concentration, because they come together, um, these two products are going to weaken the bond between hemoglobin and oxygen, so that we can help facilitate their unloading. So again, this is all in regards to just transport of oxygen. Now we can move on to the transport of carbon dioxide. It can be transported in our blood in three different ways. We can have it in our plasma, not a whole lot, but higher than oxygen. Why? Because remember, carbon dioxide has a greater solubility in water. So 7 to 10% about of carbon dioxide is transported via the plasma. About 20% is bound to hemoglobin. It doesn't bind an iron atom in the middle or a heme group or anything like that. Um, Instead, it binds an amino acid somewhere on one of the globulin chains that make up hemoglobin. So because it binds an amino acid, we call it carb amino globin. I'm sorry, I think I forgot the hemoglobin. Carb amino hemoglobin once it's bound. So 20% directly bound to hemoglobin. But the bulk of our carbon dioxide is not transported via hemoglobin like it is oxygen. It's actually transported in a different form. And the different form it's transported in is as a bicarbonate ion. So what happens is we have the bulk of our uh, carbon dioxide entering our red blood cells. Okay, we have about 90% that enter our red blood cells. 20% that enters our red blood cells binds to an amino acid on hemoglobin and becomes carbaminoglobin. The other 70% of the 90 that enter a red blood cell, because this is occurring in a red blood cell, are going to be converted to the bicarbonate ion over here. So what happens is we have carbon dioxide entering into a red blood cell, at which point it's going to combine with water. An enzyme called carbonic anhydrase is going to convert these two products into carbonic acid. Carbonic acid is really, really unstable, so it's going to want to dissociate into a hydrogen ion and then the bicarbonate ion over here. So this is all occurring in a red blood cell. Once we produce the bicarbonate ion, we don't want to keep it there, so we transport our bicarbonate ion from our inside of our red blood cell out into our plasma out here. Um, However, because we are losing a anion or a negatively charged ion from the inside of a red blood cell and sending it outside of the red blood cell, we have to replace that negative charge. Because if you remember tonicity and what happens to a red blood cell if we place it in a hypotonic solution or a hypertonic solution, we can have negative effects on the red blood cells, such as swelling, bursting, or shriveling or shrinking if we mess up the ion concentration. So we're removing a negatively charged ion, specifically the bicarbonate ion, from the interior of the red blood cell because we sent in something electrically neutral. We have to replace that negative charge. And so we do. We take chloride ions from our plasma and put it into our red blood cell. And this is what's called the chloride shift. So we swap a bicarbonate ion for a chloride ion. So now that we have and again, this is going to be in the plasma, our bicarbonate ion out into our plasma, it can travel throughout our cardiovascular system that way. However, by time when we reach it back or get it back to our lungs and we need to exhale it back out, we can't exhale out a bicarbonate ion. So we have to take our bicarbonate ion and reverse it back to carbon dioxide in the same manner, entering our red blood cell in the pulmonary capillaries before we exhale it out. So we actually have carbon dioxide diffuse into our alveoli during expiration. So again, let's review this. About 10% of carbon dioxide can be found dissolved in our plasma because it has a slightly higher solubility than oxygen itself. 90%, so if we add up these two, would be 90% of our carbon dioxide is going to enter our red blood cells. Of that 90%, 20 is going to bind to an amino acid on a heme group and become carb amino hemoglobin. The other 70% is going to react with water and an enzyme called carbonic anhydrase to form carbonic acid and dissociate into a hydrogen ion and a bicarbonate ion. The bicarbonate ion is not going to remain in the red blood cell, so it's going to diffuse out of the cell into the plasma. But because we lost that negatively charged ion from the inside of the red blood cell, we have to replace it. And we replace it with a chloride ion, and that's what's called the chloride shift. So we have our bicarbonate ion traveling throughout our cardiovascular system, heading back to our lungs. But once it gets to our lungs, we can't exhale a bicarbonate ion, so we have to go take this bicarbonate ion and put it back into a red blood cell to turn it back into carbon dioxide so that we can actually exhale it out. And the last thing, or second to last thing I need to talk about is the Haldane effect. And the Haldane effect is really just saying, let me switch, that at low partial pressures of oxygen, we're going to have low levels of oxygen bound to hemoglobin. So low levels of oxyhemoglobin. When we have low partial pressures of oxygen or low levels of oxygen bound to hemoglobin, we're more likely to bind carbon dioxide to hemoglobin. So we're more likely to make carb amino hemoglobin. So as we increase our carbon dioxide in the blood, we're going to be able to get more of the oxygen to dissociate from the hemoglobin. We didn't talk about this now, but we talked about that earlier in this lecture, and that's called the Bohr effect. So let's go back over here. The Bohr effect was as we increase the amount of carbon dioxide and increase the hydrogen ion concentration, those two things cause the bond between hemoglobin and oxygen to be weakened. So as we increase our carbon dioxide in the blood, we're weakening the bond between hemoglobin and oxygen, and now we're getting more oxygen to get disassociated from hemoglobin itself. So that's the Bohr effect. It's in play with the Haldane effect. So because now we have low levels of oxygenated hemoglobin because the high carbon dioxide caused a ton of the oxygen to dissociate from the hemoglobin. Now we have low levels of it, means that now we can make more carb aminohemoglobin. And this is the Haldane effect. So these two go hand in hand. Okay, the last thing that we absolutely need to talk about for this series of short lectures is the carbonic acid bicarbonate buffer system. I don't want to get into too much details because I know you already have a lot to cover. I just want to point out a couple of things. Anytime we're talking about a buffer system, the buffers are going to help resist changes in pH. How do they help resist changes in pH? Well, they can act as either a weak acid or a weak base. So our blood pH level should be around 7.4, so somewhere between 7.35 to 7.45. Anything outside of that pH range can have detrimental effects on us. So we have to be able to buffer our blood pH in order to keep it into that very, very, very narrow pH range. So how does it work? If for some reason we increase our hydrogen ion concentration, meaning that we've increased the acidity of our blood, which also means that we lowered the pH, what's going to happen now that we have a surplus of hydrogen ions? The hydrogen ion can come in and bind to our bicarbonate ion to get carbonic acid. In the event that we have too low numbers or concentration of hydrogen ions, meaning we've increased the basicity and the pH, now we can come in and disassociate our carbonic acid into our bicarbonate ion and deliver extra hydrogen ions so that we can increase our hydrogen ion concentration and lower the pH and increase the acidity of it. So again, just kind of understand what the carbonic acid bicarbonate buffer system is, how it can act as either an acid or a base. So if our blood pH gets too high, down here, we can add more hydrogen ions to bring it down, to bring it more acidic. And then the same thing happens is that we can pick up the extra hydrogen ions, combine it with the bicarbonate ion to form carbonic acid. So we can go pretty much both ways. And that's it. I hope this review of the different respiratory stages helps. I'm also going to post a PDF of what I just wrote on this underneath the video links.